So the last thing we're going to go over then this way is our cell respiration. So make sure that you understand the bullets underneath here, but we will diagram it just like we did up here with photosynthesis. So we know that the final product of photosynthesis was glucose. Right? We could also make any of the other macromolecules, but we're going to talk about glucose. Right? So when we use glucose, overall, the process that we're talking about is cellular respiration. Now, there's two types of cellular respiration. What are the two types? Good. So we have anaerobic and aerobic. But both of them have one thing in common. What do they both have in common? Both anaerobic and aerobic have one thing in common. That is it. Very good. So they both start with glycolysis. Right? Now, let's just make sure we're clear. What is our formula here? You definitely should know the formula. So it's essentially the opposite of photosynthesis. So you're going to take the glucose, which we just brought down, right? That's C6H12O6. What are we going to add? The last step uses what? Good. Oxidative phosphorylation uses oxygen. And we're going to get the CO2 that we breathe out and the water, and technically somewhere around 36 to 38 ATP. Oftentimes I don't put ATP on that side because if I do that, I need to put ADP and a phosphate on the other side. But you get the idea. The whole point here is to release ATP, to release energy into ATP. Okay? So for cell respiration, glycolysis happens in all living things. Oldest metabolic process, it means splitting sugar. All right? And glycolysis splits sugar into two pyruvic acids. Now, if there is, let's say, if there is no oxygen, then what process do we go to? Well, not exactly. What process do we go to at this point if there's no oxygen? Fermentation. Right? Now, fermentation and glycolysis are both considered anaerobic respiration. That makes sense to you guys? So it's both of those. Now, in glycolysis, we got 2 ATP, but what else did we get? 2 NADH. Right? Now, the problem is, if we have no oxygen, we have nowhere to drop off our hydrogens. The oxygen can't take our hydrogens. So how do we get more NADH to get more ATP? We have to use fermentation. The whole point of fermentation is that it is the regeneration of NAD+. Right? So that's the whole point of fermentation. And don't forget, there's two types. An alcoholic fermentation, which is only in fungi, you get 2 carbons and you release CO2, like the bubbles in air bubbles in bread or the bubbles in wine and beer. Right? In animals and bacteria, it's lactic acid fermentation. Right? Either way, what we're really doing is adding the hydrogens back onto it so that we can continue to do glycolysis, so that we can get some energy out of it. But if we do have oxygen, Then we can go into the rest of which process? No oxygen, anaerobic. Then we can go into aerobic respiration. All right. Now, in aerobic respiration, we take our pyruvate, which are 3 carbons, right? And the first thing that we do is pop one off. Release the CO2. And then we add something called coenzyme A to make acetyl-CoA. Now that acetyl-CoA can go into the Krebs cycle. So what happens is my 2-carbon acetyl-CoA bonds to a 4-carbon called oxaloacetic acid, OAA, to form your 6-carbon citric acid. All right, so now we have brought in our acetyl-CoA. 2 plus 4 equals 6. Now, the first thing that happens with that citric acid is we're going to pop off 1 CO2, and then we are going to go ahead and oxidize it, or we're going to take off some hydrogen and electrons and put them onto NADH. This is the important thing. Right? We're going to pop off another carbon dioxide. We're going to make another NADH. Then we're going to make an ATP. Then we're going to make an FADH2. Right? But importantly, what we're actually getting this, and this is everything times 2. We get 2 ATPs and we get all this stuff. But the important stuff that's coming out of this is NADH and F. These are our buses, which are going to take electrons over to the third step, which is oxidative phosphorylation. So oxidative implies that we use oxygen to power it, and phosphorylation implies that we're adding a phosphate to ADP to make ATP, which is chemiosmosis. Right? So in oxidative phosphorylation, these two things split up, right, into NAD plus AD plus, and they can go back. They get split into electrons, which goes to the electron transport chain, pulled by oxygen, and then the hydrogen, God bless you, forms a gradient for chemiosmosis. See photosynthesis for that, same process, all right? Then the hydrogen and the electrons and the oxygen forms water at the end. Chemiosmosis forms roughly 36 ATP. And what you end up with is ATP and water. Water's your byproduct. ATP used, heat released, energy is gone. All right, so the energy has now flowed through our system from the light up here into the glucose, and then we broke it down into ATP in many steps of the way here, and then that ATP releases it as heat.